over to Bill Amos now, who's not going to use the sound system. His voice parrot. Thank you, Bill.
I think so, yeah. Okay.
Thank you.
So I'm not sure that you really need lots of technical genetics at this point in a hot room, so I'll try to keep this to an extremely noddy level if possible. This is really sharing my experiences after severe arm injuries from the amount of twisting that Bruce exerted. I met Bruce on the Royal Society panel, which was looking at inbreeding in pedigree dogs, and afterwards he plagued me with innumerable emails saying, why don't you look at this, why don't you look at this? And I said, I've never worked on dogs in my life. And he said, no, come on, you've got to look at this. And in the end, I reneged and started what I thought was going to be a small study, but it seems to expand. So just some really basics of how you might go about looking for the gene for a nasty disease. This would be a classic pattern that you try to look for. You get a bunch of diseased animals, each of which carry 2 copies of a particular gene. And here's a bunch of healthy controls. And from the colour coding, I hope you can see by eye that the diseased ones have a lot more red compared with the healthy, and you might go away and say, oh, crikey, we found a nice association. And that all works well in very simple circumstances. But I think most of what we've heard before is that the JKD scenario isn't anything but simple, what with the skewed sex ratio, etc. And in fact, I think, well, I'm going to tell you the story of where I've got to and how I've been looking. So it's not as simple in pedigree dogs, if nothing else because of the extremely strange pedigrees that emerge. They're nothing like natural populations that I'm used to working with. Obviously you get the recycling of top studs who come into pedigrees again and again. So for example, if we consider, this is my diagram of what's meant to be the pedigree of 2 different breeders, each of whom has got a top stud dog who they've used quite a bit. And as you can see immediately that if the top stud dog here is a red and yellow, that not every gene in this pedigree is going to be red or yellow, but there are more reds and yellows in the animals produced by this breeder compared with this one over here, who has quite a few blues and yellows filtering through. Now if you just look at that pattern, you'd say, if you just treat these as random samples, you'd say these are completely different from these genetically. And it's just because they're much more related in samples from a particular breeder. And this means, for example, I've tried to be anonymous in some cases, maybe you'll recognise anyway. If we've got a number of different breeders here, and they've each got their studs and they've each got their pedigrees, maybe some of them are more reluctant to report than others, so maybe some of them don't report cases of JKD, whereas others do. So here's a perhaps worst case scenario where we've got 4 breeders and only one of them reports cases of JKD. This unfortunately means that you open up to environmental associations. So here perhaps these breeders all feed their dogs on different breeds of dog food, different types of dog food, and so here we see that breeder 3 is producing quite a few closely related dogs, so they're genetically similar, and we've got lots of JKD cases, but they've all eaten the same dog food, so you might immediately think, oh, perhaps that dog food's a problem. Equally, I've had emails from somebody who's convinced that there is a vaccine that is used, I can't remember the name of it off the top of my head, but it's suggesting that, okay, yes, okay, so a particular vaccine, and says that there's an association there. And the trouble is, statistically, many of these things are really difficult to disentangle. Many of you may remember all the stuff with MMR vaccine. It's really difficult because there you have a vaccine that is given to human children at exactly the same time as autism is diagnosed. And people, oh crikey, look, I gave my child the vaccine and it was diagnosed with autism, and they assume that there's a connection. And you can get a lot of connections which are apparent, but are actually due more to chance or spurious associations due to the structure of the data. So there's a warning there. So there are a number of possibilities for what can be the cause of JKD, and there's a whole series. Bruce has really emphasized the idea that there's one gene and that it's recessive, but there are other factors that could come in. There's inbreeding depression. We all know that there's a possible risk, particularly in pedigree dogs. There are environmental factors. It may be that you have to have a particular gene, but you only develop the disease if you get an injection, or if you're given a particular diet, or if you're stressed at a particular time in your life. It might be that there's one gene, or it might be that there's one gene plus the environment. There might be two or more genes. I float the possibility, it's slightly poo-pooed by the other speakers, I haven't thought about it much, and I'm sure they're right, but the possibility that there's a sex linkage effect, that there's a gene on one of the sex chromosomes that it sort of provides the right background on which another gene can cause the disease. And we can add more and more and more things, and really important is what Bruce was talking about, which is that there are potentially multiple causes. And it's really difficult to track down a gene when you've got half the cases have one gene and the other half have a different gene or an environmental effect, because that just muddies the waters and makes it a really fuzzy picture that's difficult to resolve. So it's really difficult if there are multiple genes or if there are environmental effects, unless you can control for this. In order to disentangle this, you really need lots and lots of samples, so that's what I've been trying to collect. And Bruce has given me a whole load from previous JKD studies. We've also had a lot of requests going out, and many of you, I think, have contributed swabs from dogs, both unaffected and affected. And a recent Facebook plea generated a whole lot more. It's particularly interesting to have affected and unaffected siblings, because in general that can control quite a lot for the environment. They will have grown up under the same circumstances, they will probably have been vaccinated in the same way, they'll have been given the same food, so if one of them gets the disease and the other one doesn't, it's much more likely to be genetic. So my sample set has an original study on tracking down brindle, the original JKD study, lots of swabs, and the Facebook plea, and this has resulted in about 100 cases and 1,100 controls. The controls are really important. You might say, why on earth are you bothering with that number of controls? But actually, the more you understand what healthy dogs look like, the easier it is to find a subtle difference that is associated with the disease. Really important is that we've got samples from all over the place. We've got lots of samples, obviously from GB, but we've also got American, French, Scandinavian, Australian, and New Zealand samples. And this is really important because the dogs are so related to each other that actually unpicking what's due to the fact they're just related rather than that the particular gene is being shared is quite difficult. So it's really nice to have as far as unrelated dogs as possible. It helps you unpick what's truly genetic and what's just simply they all come from the same breeder. So diverse lineages, large numbers, sibling pairs, these are all things that can help us try to unpick the problem. So one of the first things I did was trying to develop a fair test for inbreeding or inbreeding depression. This again it's not as easy as you might think, because there's so many, all the different breeders use slightly different versions, so the patterns of relatedness in boxer dogs are fantastically complicated. So what I did, 1, 3, oh yes, right. So for as many dogs as I have, and you would not believe the number of hours I have spent typing in pedigrees of dogs. I've got 7,500 individuals and both parents typed in. And what with the Van Ders and the VDs and the Vs and the V dot and the VD dot and the number of different ways people spell their different dog names, even though it's meant to be the same dog. Fortunately, I do some computer programming, which has helped, but I have now got a massive database, and I can type in many dogs, and my program will spew out a 5 or deeper pedigree, 5 generation or deeper pedigree. So here we have a typical pedigree just in outline. We've got a dog that may have JKD, and then we've got its parents going back, as I say, at least 5 and often many more generations. And what we want to do is to ask whether or not there's evidence that the JKD in this dog is due to inbreeding depression. Well, of course, you could select your... Oh, it seems to be doing it automatically. Or are you preempting?
It's me, I'm watching.
Oh, right, okay, I was wondering. Right, so you could just simply compare the inbreeding coefficient of JKD dogs with a whole load of healthy controls. But that doesn't really work if you think about it, because what if most of your JKD dogs come from a small number of studs which have been used extensively in a small, unusually inbred pedigree? Then they're going to be more inbred anyway, and the two factors are intertwined too deeply. So what I thought was the best control for how inbred this is relative, the best control for a dog is its parents, because its parents have come from the same breeding line and therefore will probably have experienced the same sort of patterns of breeding. So what I did was I compared the inbreeding coefficient of a JKD dog going back I've done it smaller to save having to do all these, going back 4 generations, but then I take the parents and I take them back 4 generations, and I compare the difference between the inbreeding coefficient of the average of the parents with the JKD dog. And you get a small effect, which it's significant, it's only at 5%. I want to do it on even more pedigrees, so there is a small effect, but this does support what Bruce was saying about the possibility that this is a recessive factor where you need to get it from both parents, because you're more likely to get it from both parents if you're a bit more inbred than average. So there is a very small, but just about scientifically significant effect of inbreeding. Thank you. Right, this was taken off the web and was out of focus on the web, so sorry. But this is to illustrate, thank you, dogs, for having so many chromosomes. They've got 38 pairs of normal chromosomes plus the sex chromosomes. That's a lot of chromosomes if you want to start looking for a gene. I used a very broad brush approach to start with. I used a series of genetic markers. These are bits of the DNA which have lots of different types, so you can track them easily through pedigrees. Up to about 15 states, we call them alleles. And I used just a panel of, many people would look down their nose at this, just 70 markers. 70 markers means that I can put 2 or 3 on each of the larger chromosomes and going down and maybe just 1 on each of the smaller chromosomes. But at least I mark each of the chromosomes, which will then allow me to ask whether the gene is a bit more likely to occur on that chromosome in particular. And I did have one hit, and this illustrates it. So these are our... Oh, the font has come out funny. Okay, well anyway. These are meant to be numbered. But you can see that there are a lot of them. These are the genetic states, and here are the frequencies of these states. So we've got state 1, 2, 3, 4, all these in healthy dogs. And you've got the same states in a whole bunch of affected dogs. And what you see is that they're very, very similar frequencies. So if it's common in the healthy dogs, it's common in the affected dogs, all the way through to this little group here, which all seem to be a lot commoner in the diseased dogs. And this is a significant effect. It's really quite significant. But there are a number of take-home messages, some of which are really good because they suggest that this is indicative of being close to the gene. And some of it is a bit of a pain. If you notice, there are an awful lot of these blue bars and do not differ between diseased dogs and healthy dogs. So it's not like, right, I found it, this is a 100% guaranteed genetic test. What I have is a case where there are a few of these states, a few of these alleles, which seem to be strongly indicative of being a carrier for the disease. For me, I was particularly convinced by this because, in fact, if you look at which ones seem to cause risk in different populations, they seem to be a bit different. So the US tends to have this one, whereas the British dogs seem to have this one, and the Scandinavians tend to have this one. Which is interesting, because that's exactly what you'd expect, because these markers, in this particular case, they tend to evolve by going to adjacent types. So probably these states are all related to each other if you go back a long way in time. So it's kind of what you'd expect if the gene was sitting next to an allele that had changed a little bit, but not too much. And so it sort of makes biological sense. So the positive news is this looks as if I've found a part of a chromosome which is sitting quite close to a gene. The negative bit is it's far from a magic test where you can test and you can say this dog will get the disease or this won't. A particularly interesting story was that Bruce told me about one of the top studs that was really used a lot and had produced many affected offspring. He said, this dog is homozygous. He carries 2 copies of the gene. Well, I could test this because I had 13 of his offspring in my sample set, and in fact, all of them had allele risk 13. That was one of these alleles I'd identified as indicative of potentially related to the disease. So kind of, that's quite a coincidence. It's not a common allele, and yet Bruce had said this dog is homozygous. And in fact, I then found that a historical sample, we had a sample from the dog, and he was in fact homozygous for 13.
And was he healthy or healthy?
The complication is that he was perfectly healthy, which seems to be this male-female divide, that it rather supports again the idea that Bruce was putting forward, which is that males seem a bit more tolerant of having a faulty gene, and maybe their kidneys can function on and they can push on into later life or even survive and not get kidney disease at all. But this just shows you that it's not a simple problem, and I'm afraid the negative side of my talk is that really we're looking for something which probably doesn't have a perfect solution. It's not like you can do a test for Huntington's disease in humans, and yet they know exactly what they're looking for, and if you have this particular characteristic, you will get the disease. So it's unlikely to be cut and dried, but I am optimistic that we will get closer. And in fact, this is what I've been doing recently. Here we are. It's at the end of chromosome 1, on one of the biggest chromosomes. This chromosome is about 165 million letters long in DNA terms. That's a lot of letters to look at. I've so far focused in on an area in which I started reading 3 million letters from each of my dogs to start looking to see whether or not I could find a gene in the surrounding region. This only produced results which were about similar to the one before, but I now believe, in fact, that because there was another marker that was further down here, which showed a medium strong signal, that probably the gene lies in this region here, slightly below the red line. So I'm now going off and I'm going to repeat it with another 6 million bases, but this time 6 million letters. But this time, I've gone for a region that's a little bit further down, and I've got information back on a test run of 50 dogs, and it kind of looks promising, which is good. I hoped that I'd have the results for this talk, but you do 2 weeks of experiments, you then send stuff off to China, and 40 days later you get the results back. It's quite expensive, so you do a test first to see if it's working, and then you go back and do all the dogs, and then you find you've gone to the wrong area, so you do another test on the next lot, and then and each one is a 40-day lag, so unfortunately I do get emails all the time saying, how's your research going? What are your results? And I have to say, they're still sort of brewing. But I hope to have my main analysis of this bit of the genome ready soon, and I'm optimistic that it will be a bit of a stronger result than I got before. And of course there is this, the sex bias, which I'm also starting to look into, and it's a good guide because actually it struck me that really I should be using all females for my main test because they're the ones with the disease, and that including males will kind of muddy the waters a bit because you get healthy males, as we know, who seem to carry the gene, but they're still healthy. So I think that's lots of thanks, particularly to people in the different countries who've been providing samples, and really in particular, poor old Mathieu Brocheton, who was a French guy who was fantastically enthusiastic, and he unfortunately was killed in a car accident halfway through the study. But I mean, he huge amounts of energy, and he managed to get all of his friends to send me samples, etc. And I have received some funding from the Council of Dog Breeds and the Kennel Club, which has allowed me to do this. Thank you.
Bill.